Okay, yeah. So welcome to the extra session for this particular course, NGS Data Analysis and Applications. Today, like I'll take this class for one and a half hours. So my primary objective for this class is just to cover what has been done across the 12 weeks, and you kind of give you a recap of the different topics that we've gone through. And if you have any particular topics that you want me to revisit, you can let me know as well. Yeah. Okay. So just to give a recap, we started with different type of genomic sequencing technologies. Then we looked into different types of pre-processing techniques, quality control, and how after generating the data, how you can use QC measures on the data. After that, using different pipelines, how can you uh, identify single nucleotide polymorphisms and big structural variants as well. And also after that, we talked about RNA sequencing analysis, different type of algorithms for alignment for RNA sequencing data. Then counts data and how can you convert it into TPM, FPKM, and different types of measures. And after that, we talked about differential expression, gene expression analysis using DEC2, and finally functional enrichment analysis, and at the end, uh, chromatin chromatin assembly techniques, like basically if you have different types of chromatin assembly in different parts of the genome, how that affects your gene expression. And yeah, that was all. So I'll quickly kind of go through not each and every topic, but the major topics that have been covered here to give you a recap. I think your exams are very near as well, so I think this might help you in the exams as well. And as usual, this lecture will be recorded and put on YouTube. So if you want to look at it later, you can do that as well. Okay, so we'll start with talking about sequencing technologies. Uh, Yeah. So the first sequencing technology that ever was there was the Sanger sequencing technology. This guy Fred Sanger won his second Nobel Prize for the invention of this technique in 1977, and this was one of the most widely used technologies since at least till early 2000s. So the basically principle behind this technology is basically DNA polymerase will naturally use DNTPs to synthesize a new strand. So if the synthesis process occurs very quickly, it becomes very hard to make any sort of measurements during the synthesis. So to overcome that problem, what you can do is you can terminate this particular synthesis step by step using a modified version of DNTPs known as ddNTPs, which are dideoxy nucleotide triphosphatase. And the DNA polymerase attaches to these DDNTPs and uh, like adds these DDNTPs to a sequence of DNTPs, but it cannot attach anything further after that. So it kind of brings it to a halt. And when it brings it to a halt, you can get an idea what base has been added to that particular strand. Right. So this technique is also known as sequencing by synthesis, and it takes advantage of the fact that the DNA strands, which are normally double helix form, can be split apart for mitosis, and each strand is copied. And you can use and you can use DNA polymerase and DNTPs to synthesize a new strand. And if you want to like slow it down, you just use DDNTPs instead of DNTPs, because after adding a DDNTP, there cannot be further addition of any particular basis. So uh, that's the whole concept of sequencing by synthesis. Is basically you try to synthesize one particular base, and then you stop the synthesis and check what base has been synthesized, and then you again continue. So that's the whole idea behind that. So yeah, this is a figure that kind of shows that. So you have a DNA strand, you have a whole lot of free nucleotides out of them, which a small fraction are DDNTPs. So it's kind of 1:99 fraction. Then you for synthesis, for DNA synthesis, as we know, you need to add a primer. So after addition of the primer, you can then continue the synthesis with the help of the DNA polymerase enzyme. And at every point, say for example, you have only P's as that has been labeled as DDNTPs inside your solution. And so what will happen is every possible position where there is an A here, it can be possible that the DDNTPT will come and bind and it will stop the synthesis. So for 98% of time, you'll get the whole sequence, but in some cases you'll get this truncated sequence because after that no further um, nucleotides can be added because this is a DDNTP. And what you do is after this has been stopped, you measure the length of each strand using gel electrophoresis. Basically, whichever is the highest strand will travel the lowest, and whichever will the basically the lightest strand will travel the most. And you can do this like in one step you can use T's, then in the next step you can use G, C's, and like this you can kind of iterate. And what you'll get is you'll get uh, if you do this enough number of times, you'll get this uh, halted DNA replication process at every point in the base here. So then you can have according to length you can sort these particular sequences by gel electrophoresis, and then you can have an idea about what the sequence is. So this is how Sanger sequencing works, but there are limitations to Sanger sequencing. The Sanger sequencing works for sequences roughly below 700 base pairs in length, and the read limitation stems from the fact that as the length L of a sequence increases, distinguishing between a mass of length L and a mass of length L plus 1, what you're doing during gel electrophoresis, that becomes increasingly harder. So that the tolerance of 0.1% measurement would make it impossible to distinguish a sequence of length 100 from 1,000 with a length of 1,001, even if all bases had the same molecular weight. So it's practically impossible for longer sequences to be sequenced by Sanger sequencing methods, because at a certain point of after a certain point of time, what we realize is there is not weight differences between two corresponding strands, two corresponding sequences. So to overcome that, we can come into next NGS sequencing, which basically you can sequence millions of DNA molecules simultaneously, and it's also known as high throughput sequencing, which is kind of the catch-all term used to describe a number of different modern sequencing technologies. And the high demand for low-cost sequencing has driven the development of high throughput sequencing, which produces thousands or millions of sequences at once. So uh, from Sanger sequencing, we move on to NGS sequencing, and classified this NGS sequencing has been classified into different generations, and NGS has led to the overcoming the limitations of conventional DNA sequencing methods and has found usage in a wide range of molecular biology applications as well. So the most common NGS sequencing technique that has been used very often is the Illumina sequencing technology, which is technically a short read sequencing technology. So this is actually second generation sequencing, which has been pioneered by Illumina. So this is very similar to a Sanger sequencing process in terms of sequencing by synthesis, but there are obviously differences in this. So uh, firstly, it achieves this technology you can use to sequence reads in parallel with each other. So Illumina achieves parallelization by running several synthesis experiments at once. Each of the template strands is anchored to a chip, and only the DDNTPs with fluorescent tags are available during the synthesis and not DNTPs. So you're replacing all types of DNTPs with DDNTPs. In each type of DDNTP is tagged in a way that it emits a different wavelength of color when it gets attached to the prior base or the prior nucleotide. Since DDNTP is Hall synthesis, the synthesis of new strands are synced. All new strands are of the same length at the end of each synthesis cycle, at which point a picture of the chip is taken. The picture is then analyzed by a base calling software to identify the complementary nucleotides To override the chain termination, Illumina sequencing uses reversible time termination. So basically, once you have added a DDNTP, it should ideally be stopped the sequencing. But what you can do is you can use this reversible termination technique. So the sequencing process introduces an enzyme which can turn a DDNTP into a regular DNTP after it has been bound, allowing the synthesis to occur. So in each cycle, you'll add a DDNTP, you'll take a picture of what the base is there, and then you add a enzyme which can convert this DDNTP to a DNTP so that you can continue this iteration. And for high C, which is a Illumina machine, sequencing machine, a single cycle can take about 6 minutes to occur. Okay. So yeah, so this is how for each cycle it will take a picture. So for cycle one, you take this picture. For cycle two, you'll have this picture. So whatever base is getting added for each cycle, you can have that picture like that. So other than that, there will be limitations to this type of sequencing as well. So even though specific limitations can vary from one type of Illumina sequencing to other, there are few key limitations to Illumina sequencing that are important to consider. Illumina sequencing is limited by the size of the fragments that can be sequenced. The limitation is due to the fact that the chemistry used in the sequencing process, which relies on short stretches of DNA being attached to a glass plate. Larger fragments of DNA cannot be attached so easily and therefore cannot be sequenced so accurately. So, if you are getting large DNA reads, which are long reads basically, more than 300 base pairs, then you might not want to do Illumina, you might want to do some sort of long read sequencing technique uh, we have discussed. Uh, and it can take a long time to generate the sequence data, and uh, like there is no actually real-time access to the data, so you have to wait till the whole sequencing is complete for a day or two for certain number of samples, and after only then you can use the data to to do your analysis. So this issue of short reads in Illumina is overcome by long read sequencing techniques. One of them, which is a single molecule SMRT sequencing. So SMRT is based on the sequencing by synthesis approach as well. The DNA synthesis is called so-called zero-mode waveguide (ZMWs), small well-like containers in the capturing tools located at the bottom of the well. So you have a well, you have capturing tools that are located at the bottom of it, and the sequencing is performed with the use of unmodified polymerase and fluorescently labeled nucleotides freely flowing in the solution. So in the Illumina case, we use DDNTPs, but in SMRT, what they use are fluorescently labeled nucleotides. Anyway, the usage of these kind of special molecules is for the fact that you want to have some sort of signal telling you that that particular base has been added. That's all that you want. You can do it using fluorescence. You can do it using chemically modified NTPs as well. You can think of a number of ways to do it as well. Right. Uh, uh, the wells are constructed in a way that only the fluorescence occurring by the bottom of the well is detected. The fluorescent label is detached from the nucleotide at its incorporation into the DNA strand, leaving an unmodified DNA strand. The SMRT technology allows for detection of nucleotide modification. It can be done through the observation of polymerase kinetics, and this approach will let you allow to sequence around 1,000 nucleotides. Yeah. This will allow to sequence around 1,000 nucleotides at one time. So this is basically a long read sequencer. So this is the whole workflow. So you have this is your original template DNA strand. You have a DNA polymerase which is adding on the fluorescently tagged nucleotides. And these are the nucleotides here. So uh, you have a you'll have a primer at the beginning, and then the DNA polymerase will start acting on the template to synthesize the strand. So for each uh, nucleotide addition, there comes out a fluorescent signal which is emitted, and the signal from each complementary nucleotide is read as it is added to the sequenced fragment. So one sequence one nucleotide is added, it emits a signal. Next nucleotide is added, it emits another signal. So from each complementary nucleotide, you can read the signals and find out the complementary basis of the DNA and predict the whole sequence. So another type of LRS technology that has been there along with SMRT is PacBio sequencing and the hi-fi reads from PacBio. So hi-fi reads are produced using circular consensus sequencing uh, on PacBio long read systems. Hi-fi reads provide base resolution of around 99.9% single molecule read accuracy, and they can be used across a wide range of SMRT sequencing applications, from whole genome sequencing, from to the genome assembly, to whatever you want to do with it. So you start with a high quality double-stranded DNA, and what you do is kind of circularize on the ends of this SMRT reads to form this SMRT bell libraries, and then you add primers to these areas which are like not double-stranded, and you add DNA polymerase, which will synthesize the strand. So what it does is circularize DNA sequenced in repeated phases. So when you do this, what it'll do it is break it off the middle portion, and it'll turn into a circle, which will have a whole circular double-stranded DNA. And the polymerase reads are trimmed of adapters to yield subreads. Consensus and methylation status are called from subreads as well. So this is how you get these high fidelity reads, which are actually pretty accurate, to be fair. Another type of LRS technology is ion torrent sequencing. So the, the principle here is when a complementary nucleotide is incorporated to a strand of DNA, a phosphodiester bond is formed, releasing a hydrogen ion, which changes the pH of the medium. The ion sensor will kind of get this signal, will see this signal and convert this signal to a chemical information, and convert the signal of the chemical information into digital electronic information. So as in other kinds of NGS, the input DNA or RNA is fragment around 200 base pairs. Adapters are added on, and one molecule is placed on a bead. These molecules are amplified on the bead by the emulsion PCR, and each bead is placed into a single well of slide. So, so you have a slide like these, and the slide is flooded with a single species of DNTPs along with buffers and polymerases, one NTP at a time. The pH is detected is each of the wells, as each hydrogen ion release will decrease the pH. This change in pH will allow us to determine if that base and how many thereof were added to the sequencing read. So you have a plate like this at the end, at the bottom of the well, where you have the original DNA sequence. On that, you add the NTPs and you add the polymerase. So it will add one DNTP. The polymerase will add one DNTP at a time, and after that, what will happen is after addition, there will be a release of an hydrogen ion, which will change the pH inside these wells, and this change in pH is detected by the sequencing machine, and how much change there has been to the pH will determine which base has been added, right? And after that, after this like kind of addition of these DNTPs with the help of DNA polymerases, they can be washed away, and the process can be repeated, cycling through different DNTP species. So at first, you start with only one particular DNTP species, and then you can kind of iterate over the rest of the stuff. So, uh, so here is an example. So first you add a G, which changes the pH. And then first you add a T, which changes the pH. Then you add an A, then you add a C, then you add a G. All the different additions of the nucleotide will bring about different changes in pH inside the well. So, and this pH change is detected like this. So for like for each pH change, there is a significant signal associated with it, which will tell you if it is a, T, or G, or C, whatever uh, that is. There is a particular signature of each of the nucleotides and how much pH they can change inside the well. So yeah, coming to the last part of the sequencing technologies part. So we have the last part is the ONT or the Oxford Nanopore technology. Sorry. And in this one, DNA can be sequenced by threading it through a microscopic pore in a membrane. The bases are identified by the way they affect ions flowing through the pore from one side of the membrane to another. So you have you have kind of a kind of a microscopic membrane pore like that. So you have a pore here, and that this particular pore is made up of two proteins. So one protein will unzip the DNA helix, and the second protein creates the pore in the membrane and holds an adapter molecule. So this is an adapter molecule which kind of keeps the bases in place. And depending and on top of that, ionic current is flown inside through the membrane. So the flow of current through the pore creates a current. Each base blocks the flow to a different degree, altering the current. So here also, instead of in ion turn sequencing, you have the change of pH as a signature. Here what you have is the change of current flow through the membrane present along with the DNA molecule. That will tell you how much what bases are actually present. So those also have a signature. So this is also long read technology, and you can sequence up to, like, I think 1 kb. It can start from 1 kb. You can go up to 10 kb of reads for ONT technology. Right. Okay. Now we come to uh, more nuanced topics inside this genomic data. So first is depth and coverage. So depth, what is depth? Depth is basically the number of times a particular base has been sequenced by your sequencing technology. So depth of a technology, or a depth of your experiment, or depth of your uh, NGS data is basically telling you how many times a particular base has been sequenced by the machine. How many evidences do you have against a particular base? So depth and coverage is basically, on the other hand, on an average, all the bases in the human genome, which is like around a billion, how many times on an average a single base has been sequenced? So how many times the whole uh, uh, genome, which is a billion, 3 billion in, in length, how many times the 3 billion number has been sequenced, right? So this is important in variant calling, because the higher depth and coverage you have, the better the algorithms will work, and also it helps you complete the data. So high coverage ensures that the entirety of the target region is represented in the data. If the coverage is too low, there might be gaps in the sequencing data, which can be leading to misinformation. So this is your depth of coverage. So this is given as a reference genome. So depth of coverage is given how strong a genome is covered by sequencing fragments. Per base coverage is the average number of times a base of a genome is sequenced. The coverage depth of a genome is calculated as the number of bases of all short reads that match a genome divided by the length of the genome. Right. So it's often expressed as X. So, like, in general, WGS will have around 30x coverage. So each base has been covered 30 times. And the breadth of the coverage is basically how much of the genome has been covered. Not not a single region, but how much. Are there regions that are not covered even by a single read? So the breadth of the coverage is the percentage of bases of a reference genome that are covered with a certain depth. For example, 90% of the genome is covered as 1x depth, and still 70% is covered at 5x depth. And this is possible. So after you generated the bases, then you need to assign certain quality scores to the bases, because as you were seeing, like we identify these bases on the basis of particular signals, be it fluorescence, be it changes in current across a membrane, or other changes in pHs or something. But these can be error prone, and there can be an associated probability of error with each of these particular claims. So we use something called the Q score, or the Fred quality score. uh, which is a common metric used to assess the accuracy of a sequencing platform. And this method proved to be highly accurate across a range of sequencing chemistries and instruments, making it the quality score standard for commercial sequencing technologies as well. So, what is the Fred score? So, in the context of sequencing, Fred scaled quality scores are used to represent how confident we are in the assignment of each base by the sequencer. So, it's basically nothing. So, if you your E is basically your probability of getting that particular base wrong. So, your quality score will be Q. It's basically given by Q. Q is minus 10 of log of that particular uh, error. So, uh, your Fed scaled base quality scores can range from 2 to 40, with some variations in the range depending on the origin of the sequence indicator. A higher score indicates a high probability that a particular decision is correct. Conversely, a lower score will indicate that probably that particular decision to assign that position to that particular base is incorrect. Right? So yeah, so if you want probability of error, you can just take the negative anti-log and you can get it. So this here it is in this table, it is quite clear how it works. So if you have a Fred scale of a Fred quality score of 10, so basically you substitute 10 here. So 10 to the power minus 1 is 1 by 10th, which is the 10% error, and accuracy is 90%. So if it goes on higher and higher, at 20, it's like 1% error, 99% accuracy, which is pretty good. And the more you go up, if you go to 40, it's like 99.99% accuracy. 50, 60, the number of nines just keep on increasing, right? So beyond 30, the amount of error is like very minutely different between different quality scores, right? So next we come to different type of data formats. The first and the most simple one is the FASTA format, which is uh, sorry, yeah, which is basically just a representation of a sequence of nucleotides or a sequence of proteins that is present in a particular genome or a particular protein inside a cell. So the FASTA format is uh, it's just the sequence with no quality scores present. So you cannot store anything else other than the yeah, sequence. Obviously, you can have like headings of the sequences and those stuff, but not anything else. So uh, in an ideal world, a reference genome would contain a single uninterrupted sequence of a DNA for every chromosome. While the resources from some well-studied species include a chromosomal level assemblies, which which have much sequenced organized into chromosomes in a FASTA file. Even these genome assemblies often include a large number of short fragments of sequence that are too long, that are known to belong to a particular species, but whose location and orientation in the genome will remain unknown. So you can a particular FASTA file, if you do not know the location and if you know this the sequence, FASTA is the format to use. So it's pretty straightforward. You have an arrow like this. Every heading starts like this. So you have an arrow, which is an identifier. Uh, the identifier is this number in this case, and the remainder of the line followed by white spaces with the first word are comments about the sequence. So this is, I think, the name of the species that is used, and other stuff about the sequence, right? So in this case, this number is the name of an assembled chromosome in this reference genome. The remaining lines give the DNA sequence of that assembled chromosome. So in the above fragment, this is important. In the above fragment, we see that DNA is either in uppercase or in lowercase. A common convention is that the case bases are segments of the DNA that has been inferred to include repetitive DNA. It is worth noting that if you are trying to design assays from sequence data. So also when you're doing alignment as well, so portions of the genome which have very high repeats, so it's highly likely that a particular read might go and falsely align to that particular region of the DNA, but ultimately it's just a repeat region. It's not the actual region from where the read originated. So to avoid occasions like this, we use masking. So this is what is known as soft masking, just converting the uppercase to lowercase. You can use hard masking as well, just converting this particular stretch of DNA in lowercase into n's. So n, like that, it'll go on. Okay. So most reference genomes contain gaps, and sometimes the length of these gaps can be accurately known, in which case the missing pair is replaced by an n, as I was saying. Sometimes gaps of unknown length are represented by a string of n's of some fixed length, like 100 or something. So next type of data format after FASTA, a little bit more complicated, is FASTQ, which is the Illumina format, you can say. So FASTQ format is a standard format for lightly processed data coming off an Illumina machine. A typical extension for FASTQ is .fq, and as we have seen, paired end sequencing produces two separate reads of a DNA fragment. Those two different reads are usually stored in separate files, named in such a manner as to transparently denote whether it contains sequences obtained from read 1 and read 2. So it's all FASTQ format is very similar to FASTA as well. So you have an identifier line on top, and then you have a sequence of ATGCs as we saw in the FASTA format as well. And then you have a line that is always simply a positive sign, but can be optionally followed by a repeat of the ID line. And then you have an ASCII coded Fed scale base quality score. This gives the estimated measure of certainty about each base calling the sequence. So you will have it like this. This is your identifier. This is your sequence, and after that you have a positive sign. Then you have another uh, set of ASCII characters followed by that. So this is telling you at each position what is the base quality score, and these ASCII values are encoded. So you have a value corresponding to A. You have a value corresponding to minus, less than sign. All you have a value corresponding to that. So these are just encoded variables here. So yeah, the line starts with at the date, and then you have stuff like this. The colon and the slash separators, slash are field separators. The separate parts of the line above are interpreted something along the lines as follows. So this at the rate is a mandatory character that starts the ID line. The next number after that is a unique sequencing machine ID. After that, it is given as a colon, then a 64, which is the run number on the instrument. Then you have this sequence, which is the unique flow cell identified number, on which particular flow cell was that particular stretch of DNA sequence. Then you have a lane number, tile number, x coordinate of the cluster within tile 1 and y coordinate of the cluster within tile 2, and whether the sequence is from read 1 and read 2. All these information you have inside the FASTQ file. Now we come to now we have seen how the data is getting generated. Now we come to quality control. So there can be various types of quality control. So checking that the quality of the base calls are proper above a certain value to ensure there were no issues during sequencing. Examining the reads to ensure that the quality metrics adhere to our expectations, obviously, and exploring reads for contamination. This is very common and this is very important. So the raw QC goals include identifying sequencing problems and determining whether there needs to be like resequencing, or do you need to like do it again, or will this work. Second thing is identifying over-represented contaminating sequences. Next is gaining insight into library complexity, which is like maybe there is some sort of RNA contamination or some sort of duplications, and ensuring that the organism is properly represented by the percent GC content. So there is a fixed amount of GC content that is present in individual genomes. Uh, and this varies from species to species. So in a certain in a certain species, this value will remain constant. Okay. And yeah, so all this stuff is done using FastQC. So it's a very basic program. You can download it directly from the website. You just need to provide the first few files there, and it will give you a summary with the HTML based report. So I do not have the slides here, but we talked about these different quality scores in earlier classes as well. So after that is getting quality scores and number GC content and everything. After that is done, we use we'll remove duplicates. So Picard tools is one of the major softwares that is used. This is the function. So mark duplicates hyphen hyphen to remove duplicates like this, and then you can do it using SAM tools. Marked up mark dup. So mark duplicates function as well. Now after that, after you've removed duplicates and pre-processed your data, you can also trim your data. What are you trimming? Number one is adapters, because you had added adapters while sequencing, so you need to trim them. They're not part of the genome. So this is done using softwares like adapter removal and Trimomatic. And you can also remove low quality bases. Trimming can also involve this cutting of bases from the start or end of a read if they fall below a certain quality threshold. So there is one, this Trimomatic software sliding window trimming method. It scans from the 5' end of the read and removes the 3' end. If the average quality of the group bases drop below a certain threshold. So this prevents a single weak base from causing the removal of subsequent high quality data. So that is there as well. Now we have done all kind of processing, quality control and everything. Now you want to do the major, the final steps of this analysis. So uh, first comes the alignment problem. So first you need to align this particular read information that you have to a reference genome. So what, defining what is the alignment problem is basically, say for example, two sequences, protein or DNA, you have it like this, x1, x2 up to xm, y1, y2 up to yn. So yeah, your optimal alignment of the two sequences in the form of a list of columns of the type. So if your x is this, y is this might be your optimal alignment like this. So right now there can be variations in the sequence, there can be mutations, there can be different types of mutations, insertion, deletions. These are at least single nucleotide. There can be transposition, a piece is removed and inserted somewhere else, or duplication, whatever be it. So what will happen if a single nucleotide deletion or insertion occurs in the coding portion of the genome? And what will happen if a single nucleotide substitution occurs in the coding portion of the genome? So all of that can be taken into account if you have a certain scoring scheme associated with your alignment. So you have, say for example, if you align A and A, you get a +1. If you align A with C, you get a -1. If you align A with D as well and or G as well, you get a -1. If you align A with a gap, you get a -2. So the algorithm is like you have a +1 if there is a match, which is a reward. You have a -1 where there is a mismatch, which is a penalty, and if a -2 if there is like if it is missing, if it is a gap like that. Okay, so like that you can try these two as alignments. So if you add the scores up, comes to -1, this comes to -2, which is telling you this is a better alignment to this, right? Okay, now the most basic algorithm uh, for actually using alignment is going to be a brute force algorithm. Just a second. Just a second. Yeah. Okay. So yeah, it will be a brute force search for optimal alignment, and if you're given just two sequence and you want to find the optimal alignment, the algorithm will try all possible combinations and output with the alignment with the maximum or the maximum score. But if you if the time taken is exponential there. So BLAST works in a similar type of way, in a brute force way. It works by comparing query sequences to a database of sequences to find regions of similarity. It uses a heuristic approach to search for similarities in the database, making it faster and more efficient. And it performs sequence alignment through the following steps. So in the first step, it'll like use the reads to create a lookup table or a list of words from the query sequence. This is also called seeding. So it will take the query sequence and break it into short segments, which are known as the words. So if you this is your whole sequence, it will break it down into three small nucleotide sequences, or in this case, amino acid sequences. For protein sequences, each word is usually a 3 amino acid long, and for DNA sequences, each word is usually 11 nucleotide long, and you create this lookup table. Next, you have they have a database of known sequences, and they'll search the database to find any sequences that contain the same words as the query sequence. And this is done to identify database sequences containing matching words. And then BLAST scores the similarity of the matching words. The matching words score is given by a substitution matrix, like the one that I showed you. And if it is a protein, and there are two commonly used substitution matrices for protein, one is the PAM, which is percentage accepted mutations, and one is BLOSSOM, which is block substitution matrix. And for nucleotide sequences, the scoring matrix is based on match mismatch scoring, as I was shown. The fourth step involves pairwise alignment by extending the words in both directions while counting the alignment score using the same matrix. If the score drops below a certain threshold due to differences in the sequences or mismatches, the alignment stops. The resulting aligned segment pair without gaps is called the high scoring segment pair, which is actually yeah, is going to be giving you uh, the reference from where it came from. So it is also calculates statistical significant value for each alignment. It's called the E value or the expected value. The E value represents the probability of obtaining the sequence match by random chance. A lower E value indicates the sequence match is less likely to be a result of random occurrence. Hence, the E value, the higher the level is of significance. It's very similar to a P value. Now, this is how BLAST works. Now, we want to align, or if you want to making as an algorithm, which basically, yeah, which basically will do the alignment for you. So one good idea to do that is in using indexing and using the concept of substrings as well. So it's not a new idea. Most books have an index where the word is mapped back to a page that has been found. So you have an index at the back. Yeah. You have a word. You're matching what to say, a word and a page where that particular word is found. So this type of index is called as inverted index. The word inverted applies that there is a non-inverted or forward index, and this image is basically uh, explaining the distinction between the two. So the word cow is present in document 2, 3, and 4, but in document, say for example 1, the words say the cow says no, like that. So if how you're mapping, depending on it, it can be a forward index or a reverse index. So for what we are interested in is searching the best location of the reference. We use inverted index. We set we like just refer to as an index. So to create an index for a string T, which is in this case a genome, first we need to extract the substrings of length L and record where these substrings occur, and organize a list of substrings and their coordinates and an easily accessible data structure, which is a map. So you have uh, this is your whole string, and you create substrings like these from here, and you store the substrings and also the key along with it where this particular substring is originating from. Say for example CCTAC. So this is the second substring. It is originating from position 4. It's a zero-based indexing. Yeah, so it's originating from position 4. The first substring CPTCG, it is originating from subset offset 0, which is the first position, 0th position. Now that you have created index for this particular text, you can search it. Suppose now we want to search a pattern, this pattern, which you can refer to as P, in the string t. To do this, we need to extract substrings from t. So basically, you need to find substrings from here as well and check the index to see if they are present. If they are present, extend to see if the entire substring is matching. So for example, you see this particular substring and you want to see if where this is. If it is there, you take the rest of the substring along with it, and you try to extend it in both ways and see if that particular step is matching. So, but there are limitations to substrings using shortest common substrings and stuff. And so we use, how do you consider the substrings also becomes an issue. So we use the idea of indexing with suffixes rather than with fixed substrings, right? So consider the following text. This is a long text, and what you do is add a dollar symbol at the end of this particular text, and all take out all the substrings of this particular text. By substrings here I mean suffixes, right? So all suffixes for this particular text. This dollar signs act as a delimiter and as a symbol that particular sequence has ended, like that. So these are all your going to be all your particular substrings. Now, if you want to make a suffix trie for this particular sequence, so it is much smaller than the text used before, and you add this particular dollar sign, and this is how it will look. So these are your end leaves, and this is your root from which it is originating, and the green part, this part is basically showing the entire suffix, and the blue part is the shortest suffix only containing the terminal character. So the longest suffix is going to be this, the whole string itself, and the shortest suffix is going to be just the dollar sign, right? Just give me a second. Hello? Yes, ma'am. These are, ma'am, I have not done that, but these results are from Mac 3 only. Um, that is possible actually. Yeah, okay, I'll do it. Mm-hmm. Okay. Oh, okay, okay, ma'am. Okay, okay, okay. Huh. Okay, 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 okay. Yeah, yeah. Sorry for the interruption in the call. Okay. Yeah. So this is your longest path, which is the entire substring, and this is your shortest, just the dollar symbol. Now, if you did not add a dollar at the end, the difference that without the dollar, the trie will not have every suffix to be represented by a path from a root to a list, right? Because like if you do not have a dollar here, so a lot of these strings will just be overlapping strings like these with one change like this. So if you do not have the dollar sign, not every suffix will be present in that path from the root to leaf. Right. So in suffix trie, every edge is labeled with a single character, and the nodes have no labels in our representation. However, you can think of each node as being labeled with a string that spells out all characters occurring along a path from the root up to that node. For example, this blue node spells BAA along a path from the root. Right. So this is the path BAA. So this node will spell this path along the root. Okay. And now, suffix trie is an ideal data structure to quickly check if a pattern is present in a text or not. Suppose we want to check a substring BAA is present within that particular text represented in your suffix trie. So this is the suffix trie for that particular text. And with, we are going to start at the root. Here we start and we are trying to find if BAA is present in this particular trie. So we take a path which is basically starting from A. starting from B. We next proceed to a path labeled as A. Then again we proceed to a path that is labeled as A. Because this entire substring BAA can be spelled out as a path from the root, we can conclude that BAA is indeed a substring of this particular text. Now, a trie is basically a branching tree. If you collapse all non-branching paths and concatenate the labels, so basically you collapse these A dollar into one node and you concatenate the labels, what you guess is a suffix tree, which is on the right hand side. Now, if you want to look for substrings in a suffix tree, uh, the logic is exactly the same as suffix tries, except now we have to account for the fact that some ages will only have a portion of characters within a label that will match. In this case, matching characters are highlighted in uppercase. So BA and 1A is getting, you can get from here. So this is not a complete match, this is a partial match. So this can happen in a suffix trie, suffix tree, not a suffix trie. And BA matches with the first edge and A matches only with the second edge, like that. Okay. Now, the main application comes is to find the longest common substring. To find the longest common substring of X and Y, if you have two sequences like this, X and Y, how do you find that? Is to build a suffix tree for X with a delimiter, Y, and again a delimiter. So this is how it looks, X, then a delimiter, then your Y, another delimiter. Now, if you build a suffix tree uh, for this particular sequence, and you can consider the leaves of offsets in 0 and 4 are suffixes of X, right? So 0 in this one, 0 here, and 4 here are all suffixes of X. So this is XY, this is XY, and this is your like basically second part of it. So offsets of these. So this part and this part has X. Right. Uh, Traverse through the tree and annotate each node according to whether the leaves below it included suffixes of X or Y or both. The deepest node annotated with both X and Y has the LCS, which is the longest common substring. The deepest node is the node which is most closest to the leaf of the tree, and that has LCS as it labeled, and it uses a linear time and space, so which is actually pretty good. Another type of data structure used for indexing and matching is basically suffix arrays. It's a similar concept. You, as with a suffix tree, you have a dollar added to it, and you have an array like that, which tells you the array tells you basically the location and the string that is there. So if this is an m length of particular sequence, this suffix array will have n plus 1 integers. So because one does not need additional pointers required for tree representation, the suffix array has a significant smaller memory footprint. For example, a suffix array for the human genome will occupy 12 GB. If you do that in a suffix tree, it will occupy 47 GB. So the drop off is massive. So yet there can be better ideas like Burroughs-Wheeler transform. So it is a reversible permutation of a string. So if you have this string, you add a delimiter at the end and you create all rotations. And you take the last column, which is the which will be the BWT of the original string. So if you have a string like this, the last column here is the BWT of your original string. So how do you get it back? First let's create all the rotations here. So this is your uh, sequence, and then you add a delimiter and create all the rotations like these, and this is your Burroughs-Wheeler transform. It has three important features that make it ideas for creating searchable compact representations of genomic data. First of all, it can be compressed, it can be reversed to reconstruct the original string, and that it can be used as an index. So how can you make it get it back? So you label it. So first, so A0, B0, A1, A2, B1, A3, and all. So this ranking is done based on the order of the characters in the text, which is T ranking, and the order of the ranked characters is preserved in the first column and the last column. So you have this order A3, A1, A2, A0, like that. And this order is followed in the first and the last column as well, right? So if you have a Balozula transform like this, so you can rank the characters in orders, ascending orders of their appearance in sorted list rotations. So this is your first column, this is your last column, and then you sort it. This is how it looks like. So A0, B0, B1, A1, dollar, A2, and A3. So what you need to find is basically what which rows are actually present with that particular characters. So when you're trying to reverse the Barzulia transform, starting on the right hand side of T and moving left. So this is your first column, this is your last column. If you just sort your last column, you can get your first column, right? So this is your first and this is your last. So you start from here, go to A0. Where is the next A0? A0 is here. You go to B0. Where is next B0? B0 is here. You go to A2. Where is the next A2? A2 is here. You go to A1, like that. So start in the first row. F must have the dollar sign. L contains the character just prior to the dollar, which is A0. And then you can do it for all the characters there, and you get the whole string back. So this is how it works, right? A0, where is the next A0, then B0, where is the next B0, then A2, where is the next A2, right? Like this. Now, after you have done alignment using whatever algorithm you want to, BWT or suffix array, suffix tree, whatever it is, what you get is an output in the format of a SAM file. So it's a text format used for storing sequence data in a series of tab delimited ASCII columns. Most often it is generated as a human readable version of its system format BAM. So BAM is basically a binary coded file of the SAM file. It's the same file, just it's written in binary. It stores the same data in a compressed indexed binary form. Most SAM format data is output from aligners, then read FAST2 files and assign the sequences to a position with respect to a known reference genome. Uh, yeah, so what information is there in SAM and BAM? The header section contain information about the file and additional information for alignments. The alignments then associate themselves with specific header information as well. The alignment section contains the information for each sequence about where, how it aligns to the reference genome. So yeah, I'll skip these. The file formats. Sorry. Yeah, this is how our example SAM file looks like. So these are all your so in alignment one, this is the number, which is the key name. Then you have the flag, then you have the R name, then you have the position, map quality, cigar, and the rest of the elements present in the BAM file, SAM file. Sorry. Now, after alignment, what do you want to do is get get to see what the variants are present in your genome, right? Yeah, what the variants are present in your genome. Now, what are variants? Variants are basically differences in the DNA sequence compared from one person to another. So it's like around 99.6 are identical and 0.4% are different. These are the genomic variants which actually give us individual characteristics and differences between individuals. Variant calling is the process by which we identify these variants from sequence data, carry out whole genome or whole exome sequencing sequencing to create these BAM files. You then align the sequences to a reference genome, creating these BAM or CAM files, and then you identify where the reads differ from the reference genome, and you write it to a VCF file, which is the variant calling format file. Right. So different types of genomic variants. I'll go over this quickly. So you have SNVs where there is a single nucleotide change. You can have insertions, small sequences inserted in a particular genome. You can have deletions. You can have repeat regions as well. So these mutations can be of two types, germline or somatic. So germline is basically mutated in your germline, which can be propagated to your next generations. And somatic mutations are acquired during lifetime, but they're not passed on to the next generation. So uh, won't affect your next generation. So uh, the considerations we need to keep in mind while detecting variants. So uh, it's it, it's it's basically important uh, to keep a few things in mind. So first is the linear isolation and fragmentation. How you do that has systematic effects on representation of different genomic regions and as a consequence on variant detection. And PCR duplicates, which is actually pretty important, which are redundant reads originating from the same DNA fragment introduced during PCA amplification of a library. And this can be common artifacts created during genomic sequencing. Um, this can falsely increase or decrease your allele frequencies or even produce erroneous mutation calls as well. Genome coverage, how much the genome is covered, is directly proportional on the number of variants you are going to uncover, and also the read length and the platform used. So how big is your reads and what platform you have used. So these are some of your these caveats that are there. So you have these PCR duplicates here, uh, which are which are present in your reads. You can directly identify them. You can see there's the coverage non-uniformity here. Some of it is higher, some of it is lower, and so on. Right? Uh, okay, so this is the most common pipeline that is used for processing reads. So you start from raw reads, then you do alignment, then you mark duplicates, you do base quality score calibration, and then you do processing of the reads. So around indels, there is something called local realignment that we perform around indels. Sorry, yeah, and it can be done using GATK indel realigner. The inclusion of realignment stage in a variant calling pipeline depends on the variant caller used downstream. This stage might be of value when using a non-haplotype aware variant caller like GATK 5 genotype or 4. However, this tool uh, for variant calling is haplotype aware like platypus 5 or haplotype caller 7. There is no need recommended to do its realignment by itself only. Then the sorting and removing duplicates is you can do is using SAM tools. Uh, Basically sorting based on chromosome and number of coordinates, and then you removing the duplicates and kind of printing basic stats. This is an important step, which is the BQSR, base quality score recalibration. It is a process by which we apply machine learning to model the errors. Model the errors systematically. Yeah. For example, we can identify that for a given run, whenever we called two A nucleotides in a row, the next base we called at a 1% higher rate of error. So any base call that comes after two A's in a read should have its quality score reduced by 1%. So we can do that over several different covariates, mainly sequence context and position in read or cycle in a way that it is additive. So the same base may have its quality score increased for one reason and decreased for other. So this can happen. And now we come to structural variants, which are basically variation in the genome which are greater than 50 base pairs in length. They can be copy number variant or they can be copy neutral. So in copy number, variation includes deletion, duplication, and copy neutral variations include inversion, translocation, stuff like that. So how do you detect SVs? There can be different types of mechanisms. One is paired read, so you have Illumina paired read data, and you know before sequencing how far were these read pairs in the original genome, and then you can see after assembly how during your uh, alignment how far they are actually inside the alignment. So there can be discordant distance between paired reads. There can be increased density of suddenly the coverage has increased, which means there might be a duplication. If there's split read mapping, a part of a read is mapping somewhere and another part is mapping somewhere else, which can be indicative of translocation. And uh, there can be other ways to determine as well. So read depth, split read, and pair read are most commonly used techniques. You can use de novo techniques as well using graphs to model this particular SVC. Now we come to the VCF format, which is basically the format in which the variants are stored. So it's a basically a tab delimited file with these are the columns: chromosome position ID, def alt, which is and the quality score, filter, and your info. This contains allele counts, allele frequencies, and other stuff as well. The format is given in genotype, and each sample will have a particular genotype. So this is a single variant calling format file. There can be like multiple VCFs file merged into one. So same position and same variant, just your number of samples will increase, giving you the genotype of that particular area. So I'll not go into the details here, but yeah, after that you can annotate the variants using ANOVAR. So according to genes, according to regions, or according to different filters present in databases. So yeah, simple and complex structural variants. So simple are just insertion, deletion, duplication, whatever. Complex is combination of both. So there is a deletion of and duplication both happening. Deletion also happening, inversion also happening. So like that. And yeah, so these are your major ways by which structural variants are kind of attained, called, and these are your different algorithms, and these are the methods used there. So RC is basically read count or read depth, and RP is read pair, SR is split read, and obviously de novo assemblies as well. Okay, I'll not go into details here. Now we come to the transcriptomic part. So instead of DNA, we use RNA to to uh, like see how the gene expression is changing inside the cell, and use that as functional data for further analysis. So yeah, this is one technique where you do gene expression analysis with a microarray, but in this case, you need to have the chips uh, made in such a way that you can capture those particular mRNAs and kind of check the counts. Right. And yeah, so if you see here, you can have different types of short read, long read, and direct RNA-seq analysis. The complexity of information captured increases from short read cDNA to long read methods that directly sequence isoforms. In short read cDNA sequencing, a significant portion of the read map ambiguously when an exon is shared between isoforms. Reads that span exon-exon junctions can be used to improve isoform analysis, but can also be mapped ambiguously when a junction is shared between these isoforms. So yeah, you have ambiguous isoforms like these, which are like kind of similar positions, but same exon being shared by multiple mRNAs. And yeah, you have you can have unambiguous isoform sequences as well. So where you don't care about the same isoform being attached to different mRNAs, you just take one and go on with it. So, but these methods mostly rely on complementary DNA conversion, which basically converts the RNA to a DNA, complementary DNA using reverse transcriptase enzymes, and then it is sequenced. But using doing that, it removes information about RNA-based modifications and can only make crude estimates of polyadenylation tail length. Direct RNA-seq enables full-length isoform analysis, base modification detection, and poly length estimation. Right? So these are your standard versus non-standard RNA sequencing protocol. In the standard protocol differs from the non-standard in two. Yeah, it separate. It is different in two ways. First, during cDNA synthesis, the second strand synthesis continues as a normal, except the nucleotide mix includes DUTPs instead of DDTPs. And after that, the library preparation on a second strand digestion step is added. So, uh, st- in standard ones, so you need to digest one of the strands and sequence the other one. Right? So you need to digest that part of the strand. So using certain type of enzymes, and this step will ensure that only the first strand survives in the subsequent PCR amplification step, and hence the strand information of the libraries. Now, after you do that, the alignment of RNA-seq is slightly different than compared with DNA sequencing alignment protocols. So here you need either your sequence aligner should be splice aware. So unspliced reads are mapped to locate exons with bowtie, and unmapped reads are then split and aligned independently to identify exon junctions. So this is an extra step that you need to do. So this is your starred alignment strategy. Majorly two processes, which is the seed searching. Next is the clustering, stitching, and scoring. For every read, star will search for the longest sequence that exactly matches one or more locations in the reference genome. These longest matching sequences are called maximal mappable prefixes, or MMPs. So a part of the read is mapping to the reference genome. There will be an unmarked portion of the reads as well. So star will then search again only for the unmapped portion for the read to find its next longest sequence that exactly matches the reference genome or the next MMP, which is C2. The sequential searching of only the unmapped portion of reads underlie efficiency of the star algorithm. It then uses uncomplex suffix arrays to search all the MMPs, and this allows for quick searching against even largest reference genome. Other slow aligners use algorithms that often search for the entire read sequence before splitting reads and performing iterative rounds of mapping. If it does not find an exact match, for each part of the read due to mismatches or indels or the previous MMPs, it will be extended. Like, so for example, you have a mismatch here, and what you try to do is like you do not only consider this portion, you consider all the rest of the position and try to see if there is a match, considering there can be indels or other type of mutations. If the extension does not give a good alignment, the poor quality or the adapted sequence will be soft clipped. So this part will be soft clipped, and this will be tried to uh, map it to the reference genome. Then the last part is the clustering, stitching, and scoring, and with the separate seeds are stitched together to create a complete read by first clustering the seeds together based on proximity to a set of anchor seeds or seeds that are not multi-mapping. Then seeds are stitched together based on the best alignment read, based on mismatches and indels, gaps, etc., and they're stitched together. Okay, so that part comes under alignment of RNA sequencing data. Yeah. Now, if you want to do de novo assembly, so you can take raw reads and kind of uh, like try to do a de novo assembly. A transcriptome constructed from short read RNA sequencing is an easily attainable proxy catalog of protein coding genes when the genome assembly is unnecessary, expensive, or too difficult. In the absence of sequence genome to guide the reconstruction process, the transcriptome must be assembled de novo, only using information available from the reads. Subsequently, the sequences must be annotated in order to identify sequence intrinsic and evolutionary features in them, for example, protein coding regions and etc. etc. Although straightforward at first glance, de novo transcriptome assembly and annotation can quickly prove to be a challenging undertaking because the amount of information you have, the amount of data you have is very limited. So this is a graph-based approach to de novo transcription assembly. So reads are divided into subsequences, which are the k-mers. The k-mers from one read are mapped like this, and k-mers build nodes on the graphs if they overlap by k minus one nucleotides, and then you form a whole graph of the reads, and then you do a work on the read to get the whole sequence. We did a similar kind of thing for DNA sequence assembly as well. So you have overlaps, you create that overlap graph, graph and you build a De Bruijn graph, and then you do an Eulerian work on the graph to get the longest sequence. Now comes the quantification part of transcripts. Generally, we quantify it by the number of reads a particular transcript has, sorry, number of counts a particular transcript has. In case of RNA-seq, the features are typically genes where each gene is considered here to be the union of its exons. One may also consider each exon as a feature in order to check for alternative splicing. Special care must be taken to decide how we deal with reads that overlap more than one feature. And you can do it using HT-seq. The three overlap resolution modes of HT-seq count work as follows. For each position I in the read, set Si is defined as a set of all features overlapping at position I. Then consider the set S, which is will be the first is the union of all the set I for all. Then intersection for sets I for mode intersection strict, and for mode intersection non-empty, it's intersection of all non-empty sets of Si. So yeah, if a gene contains precisely one feature, then the read is counted for this particular feature. If it contains more than one feature, the read is counted as ambiguous. And if S is empty, then the read is counted as no feature. So here it is only present in one gene, so it's only gene A. If it is present in both genes, if you take a union, sorry, if it is present in only one gene, but it is spanning your intron as well, if you take union, it will get you gene A. If you take strict intersection, it will give you no feature. Like that, you can do it for all the types. Then we come to normalization, and there can be two types of normalization, which is within sample and between sample. So uh, the most broadly studied methods are FPKM and RPKM. So these correct for bias of gene lengths and the number of feeds, total number of feeds, right? So yeah, this is your formula for FPKM or RPKM, where basically your raw number of feeds divided by the length of the gene, which is the feature, which is given in kilo, so it's 10^3, and QJ is basically the total number of leads mapped, so divided by 20/6, where it's basically per million. Okay. So TPM is similar. I'm not going into details here. And between sample normalization, it's basically essential for differential gene expression analysis, because your samples inter-sample normalization should be there to compare expression between two particular samples, right? So one such intersample normalization or intersample bias removal is batch effect. So batch effect is basically clustering of certain samples together, not by biological influence, but by technical or uh, sequencing errors or something of that sort. So if you have kind of uh, like two samples having which are controlled, two samples which are case. So the case samples should cluster together and the control samples should cluster together. But it might not happen due to sequencing errors or due to batch effect. So that can be done. That can be removed using lemma, which is a two-way ANOVA approach. And that you can use empirical bias approach using compat as well. So these are basically your regression modules, which kind of scale your data up and down depending on how much batch effect there is. And after that, you do downstream analysis. So as I was saying, your your control or your case samples should be together like it is here. So here is no batch effect. Right. So uh, you can do clustering, you can do sample correlation, hierarchical clustering, and principal component analysis. And so if you want to find, say, particularly if two genes are separate, like are having different expression or not, you can do student t-tests for each gene. So difference between group means divided by the variability of the groups. So this is your p-value, and along that you'll get a p-value telling you if it is significant or not. Now, the differential expression analysis is basically to check how much there is deviation in expression of a of a particular gene or a particular set of genes in one set of samples compared to the other. Right? So here there is no significant difference, but here there is a significant difference. Right? So if you compare the means as well, there is significant differences. So this is your whole algorithm. So you do unsupervised clustering analysis, then you model raw counts for each gene. In that, you estimate size factors, gene-wise dispersions, you fit a curve to gene-wise dispersion estimates, and you find a GLM, generalized linear model fit for each gene. And then you do shrinking of the log to fold changes, and then you check for differential expression analysis. So in the end, you have during during implementing this, you need a design formula. Here it is age plus treatment and sex. So since this is not separate, they have combined it into one and given it like this. So this is your design formula. So according to this, the differential expression will be calculated. And after that, you have calculated using different kind of models and like LRT or wall test, likelihood ratio test or wall test. But you cannot use the p-value directly because there are multiple tests occurring. So then you need to basically correct for these p-values. So each p-value is the result of a single test. The more genes we test, the more we inflate the false positive rate. This is a multiple testing problem. You can kind of go over it or like overcome it using some sort of correction. One-for-one correction is one of them. So it's basically adjusted p-value will be p-value times the total number of tests. This is another very useful method, which is the Benjamin-Hochberg method. Uh, you can control the expected FDR below a specified level, given a list of independent p-values as well. So if your FDR is 0.05, what does it mean? It means that the proportion of false positive we expect among our differentially expressed genes is 5%. For example, if you call 500 genes as differentially expressed with an FDR cutoff of 0.05, you expect 25 of them to be false positives. And coming to the last part, which is the functional enrichment analysis, is basically like quantifying how much a pathway or how much or a disease state is getting affected due to expression of particular genes. So you have popular tools, GSA and NHR, and these are like pretty popular. And so yeah, so basically GSA, you have to give a gene set on top of your RNA-seq data. It calculates it ranks the gene and calculates the enrichment score for all the total genes together and gives you a score telling you how much a pathway or a particular disease has been affected. Okay, not go with this. So just give you an overview of GSA. So this is in the first step. It ranks the genes based on correlation between the expression and class distinction. It computes the cumulative sum over all ranked genes, increase the sum when gene in S, and otherwise it decreases. So it goes through all the genes. If the gene is particularly present in the list, it increases the sum, otherwise it continues. And it records the maximum deviation from zero as the enrichment score. Right? So it is increasing here till here it is increasing, then it is dropping off. So here this is your enrichment score. So these are three examples where basically it is showing locations of maximum enrichment score with zero correlation between expression and phenotype. So here you have two enrichment scores here. Here it is one. Here also it is two. So yeah. So these are different pathways. This is a pathway. This is a pathway. This is another sort of set of genes involved here. So in step 2, it assesses the significance, nominal p-value. It permutes phenotype labels 1,000 times, computes enrichment scores for each of these permutations, and compares the enrichment score for actual data to distribution for enrichment scores from permuted data. Permuting the phenotype labels instead of the genes maintains the complex correlation structure of the gene expression data. So after even permuting, if a particular gene is coming up, if a particular say for example the enrichment score is not changed, that means that is your enrichment score. Then what it does is normalizes for for the enrichment score, accounting for size of each gene set, which yields the normalized enrichment score, and then controls proportion of false positive by calculating FTR to each normalized enrichment score by comparing tails of the observed and null distribution for the NEs. And in the final step, the original method used equal weights for each gene. The revised method weights each gene according to the correlation of the phenotype. And this may cause an asymmetric distribution of the enrichment scores if there is a big difference in the number of genes highly correlated to each phenotype. Consequently, the above algorithm is performed twice, one for positively scoring gene sets and one for negatively scoring gene sets. So, so this part I have completed early, with the earlier week also only. This is genome assembly part, so I'll not go over it in very detail, but yeah, just uh, let me go for overlap graphs. Yeah, so this is your procedure for making De Bruijn or overlap graphs for genome assembly. So you assume a k-mer and you pick a substance k of length something, and then you start with a input string and take each k-mer and split it into right and left k minus 1 mers. And so basically this is your k equal to 5 mer. This is your k minus 1 left mer. This is your k minus 1 right mer. And then you add each to a node and then associate it with the next node with a with an edge and create the whole graph. Now, after you've created the graph, sorry, after you've created the graph, what you do is a Eulerian walk. You start from this particular node, which is the starting node, and then you kind of try all these paths and check which is giving the best possible assembly. And you have to use an Eulerian walk for that. I discussed Eulerian walk last time. So you have to use an Eulerian walk for that, and you can get back the whole genome, right? And in the final part here, it's basically the chromosomal organization that we talked about in the actually the last class only. So you have this chromatin uh, and chromatin modifying enzymes as well. So these are your enzymes which modify your chromatin. This chromatin modification is done through DNA methylation and demethylation and also acetylation, deacetylation. And this is one technique to identify different type of methylation patterns inside the data, inside your genome. So, which is known as reduced representation bisulfite sequencing. So you purify your DNA and you use restriction enzyme to digest it. You do end repair, which is basically just extending those last parts, and add adapters. After that, you kind of let it run on a gel electrophoresis. Then you select the size and you do bisulfite conversion. Is basically where there are uh, unmethylated cytosine, it will be deaminated to uracil. Other than that, if it is a methylated cytosine, it will remain there. So using that and using the electrophoresis, you can identify at what positions the base has been methylated. Okay, I think that's all from my side. I think I have given you an overview about almost the whole course. I've missed out on the Linux parts and some hands-on R parts as well, but yeah, that I think you can take up on your own as well. And meaning like yeah, you can do it as on your own as well. But I think I have given you the major overview of the course. Yeah, that's all from my side. If you have any questions, you might ask. Okay, so we don't have any questions. So best of luck for the exam. And if you have any doubts, you might want to mail me. I'll try to help you. You can mail me here if you have any particular doubts. And yeah, best of luck for your exam, and thank you for joining the classes and attending the TA sessions. Thank you and best of luck. I'm closing this session.